I'm going to ask Jenny Wang to come up and give our first talk to you this afternoon. So Jenny is head of the Cancer and Stem Cell Laboratory at the University of New South Wales in Australia. And she's been heading a group there since 2011. She's trained with Scott Armstrong at Harvard way back in the day. So seems like a long time ago now, but she's going to be, her research really has focused on the role of G protein coupled receptors in leukemia stem cells. And this is a key problem actually in many of these blood malignancies, is you have this problem with relapses in chemo and radiotherapy, and this has been identified back to this key niche of cells, the cancer stem cells. So Jenny's going to tell us a little bit about that, and then we'll have a few questions at the end. Over to you, Jenny.
Thank you, Andy, for the invitation. I really appreciate the opportunity. I'm going to introduce myself a little bit. So I had my PhD at the Macquarie University in Sydney, Australia, and postdoc training at Harvard. Currently, I'm head of the Cancer and Stem Cell Biology lab at the University of New South Wales in Sydney, Australia. So my lab is interested in understanding the leukemia stem cell biology for developing the targeted therapy in acute myeloid leukemia. So we are working on signal pathways, metabolism, epigenetic, and non-coding RNAs in regulation of leukemia stem cells. So acute myeloid leukemia has very poor outcome with a 5-year survival rate of only 27%. And the majority of AML patients will relapse. And also you can see from this figure, you can see the incidence and the mortality rate of acute myeloid leukemia has not improved in the past 30 years. So majority of the AML patients will relapse, and the major cause of relapse is leukemia stem cells. So indeed, there is urgent need to develop leukemia stem cell targeted therapies for acute myeloid leukemia. So leukemia stem cells has two major features, self-renew, unlimited self-renew, and quiescence. The quiescence will help leukemia stem cells to be resistant to the chemotherapy. And after the treatment, the surviving leukemia stem cells can regenerate a more aggressive leukemia with highly unlimited self-renew capacity. So if we can develop leukemia stem cell targeted therapies to specifically eliminate leukemia stem cells, we can potentially develop this curative treatment for acute myeloid leukemia. So this figure is adapted from the New England Journal of Medicine, which introduced our disease model here, published in Science. So we have previously introduced demonstrated this beta-catenin signaling as a critical self-renew pathway to drive the leukemia stem cells in acute myeloid leukemia. And also the New England Journal of Medicine highlights the clinical implication of our finding for human AML treatment. So importantly, the beta-catenin signaling is not only critical for leukemia stem cells, but also very important for cancer stem cells in solid tumors, including lung cancer, breast cancer, colon cancer, and prostate cancer, among others. So how are we going to target beta-catenin signaling? Unfortunately, beta-catenin itself is not druggable, mainly due to the localization and the structure of this protein. So if we want to block this signal pathway here, So we have to target, we can potentially target the upstream components of this signal pathway. So a couple of years ago, we collaborated with Novartis to test this drug called XAV939. So this drug can degrade beta-catenin by stabilizing this beta-catenin destruction complex. However, we could not find a drug effect on leukemic stem cells. We also tested a number of the Wnt beta-catenin inhibitors, and also we could not find any drug effect on leukemic stem cells. So this brings us to the question whether or not normal and leukemic stem cells use the different upstream pathways. Here. So as you know, in the normal leukemic stem cells, the Wnt ligand can activate beta-catenin signaling. This is a typical pathway for Wnt signaling. However, in the leukemic stem cells, there are no mutations identified in the major components of this pathway. And also there's no increase in Wnt signaling in the leukemic stem cells. So those observations suggest even though beta-catenin is activated in normal and leukemic stem cells, it may regulate different signal components. So this also explains the treatment failure for the Wnt inhibitors in the leukemic stem cells. So this also implicates other signal pathways might involved in activation of beta-catenin in the leukemic stem cells. For example, G protein coupled receptors. So our studies and other publications suggest that these G protein coupled receptors called prostaglandin E2 receptor, EP2, can regulate beta-catenin signaling. So basically, if you treat leukemic stem cells with this PGE2 inhibitor called indomethacin, you would observe the growth inhibition of leukemic stem cells in both acute and chronic myeloid leukemia. So we found that indomethacin can suppress beta-catenin signaling by block this signal pathway, and also indomethacin can efficiently block leukemic stem cell initiation. However, indomethacin has less effect on the fully established leukemia. So it's very important to identify G protein coupled receptors, which are critical for leukemic stem cell maintenance, because AML patients, they have this fully established leukemia. So if we wanted to develop a curative treatment, we do have to target those fully established leukemia. So here we go. So we identified this G protein coupled receptor called GPR84. So GPR84 is another inflammatory receptor, and the GPR84 is a key driver of beta-catenin signaling for the leukemic stem cell maintenance. Interestingly, we also found that GPR84 can regulate leukemic stem cell metabolism through this G-alpha-Q signaling. So in the next few slides, I will give you some evidence to support the role for GPR84 in the maintenance of leukemic stem cells. So from my current result, we found that GPR84 is preferentially overexpressed in murine leukemic stem cells, and also in the patient leukemic stem cells compared to normal stem cells. So we also looked into AML patient database, and we found that upregulation of GPR84 is correlated to poor survival in AML patients. So this is consistent with our animal study. So genetic knockdown of GPR84 can prolong the mouse survival. So this gives the strong evidence suggests that GPR84 is responsible for the leukemia maintenance. So here I give you the evidence to suggest that beta-catenin is a functional downstream target of GPRD4. So overexpression of GPRD4 can activate this beta-catenin signal, the beta-catenin associate gene signature in leukemic stem cells. And so, as I mentioned before, the genetic knockdown of GPRD4 can prolong the mouse survival, induce this GPRD4 deficient phenotype in the animal model. So if we retrovirally introduce this active form of beta-catenin into the GPRD4 deficient leukemic cells and put those cells back into the mice, as you can see, this overexpression of beta-catenin can rescue the GPRD4 deficient phenotype in this animal model. So this provides evidence suggests that beta-catenin is a functional downstream target of GPRD4, and GPRD4 is a promising new therapeutic target for AML treatment. So how are we going to target the GPRD4 signaling? So we are currently collaborating with this French biotech company, this is a clinical stage biotech company, to test this preclinical GPRD4 inhibitor for the AML treatment. So this inhibitor has been used for the phase 2 clinical trial, so have demonstrated the safety and targeted engagement. So we found that GPRD4 inhibitor has this potent anti-leukemic stem cell effects for the AML treatment. So summarize my talk, so we found that these different G protein-coupled receptors are required at different stages of leukemic stem cell development. So GP2 signaling is required for leukemia initiation, and GPRD4 is required for leukemia stem cell maintenance. If we want to prevent leukemia, we can target the GP2 signaling. If we wanted to develop the curative treatment, we can target on the GPRD4 signaling. So we also know the G protein coupled receptor can regulate the multiple function of leukemic stem cells through this crosstalk with other oncogenic pathways, for example, metabolic pathway, epigenetic pathway, and non-coding RNAs. So we are currently working on the pathway crosstalk to understand the complexity of leukemic stem cell biology. So this is a key step on the path to the personalized medicine. So we know for the individual patients, they have very complex the genetic background. So if we can understand the complexity of leukemic stem cells, we can potentially develop a more effective and more specific leukemia stem cell targeted therapy for the personalized medicine. So we are currently collaborating with several biotech companies to develop novel leukemia stem cell targeted therapies. We have already established this powerful disease platform to understand the complex cancer biology and to develop innovative leukemia stem cell targeted therapies. So here we are open to the new collaboration with industry partners. Before I finish my talk, I would like to thank my group and also the funding bodies to support my lab, and also collaborators and the biotech companies. Thank you very much for your time and attention. I'm ready to take some questions.
Great, thank you very much, Jenny. So what we're going to do, because this is quite a challenging environment, we're going to leave the questions until the end of each session. So if you have questions for Jenny, I would suggest that you approach her like at the end of the session. In the meantime, so one question that plagues all cancer stem cell research at the moment is, in many ways, similar to many other areas of new biology. So there was a great deal of excitement about cancer stem cells. A few years ago, there was a company called Oncomed that GSK spent billions of dollars buying, and then nothing came out of it. So I thought the data that you showed about the Wnt pathway and the mutations that just are not seen in the patients populations that you're looking in is quite informative. So could you just kind of give the audience your thoughts as to, you know, how you see this area in terms of the hype that maybe was about cancer stem cells before, and where you see the real promise of the therapies? Is it going to be for blood malignancies? Do you think it's also a possibility for solid tumors as well?
Yeah.
So just a few words on that, Jen.
Yes, certainly. I would think this is certainly going to apply for the different cancers. So as we all know, the Wnt beta-catenin signal pathway is really very key pathway for the cancer stem cells. So, and a number of biotech companies actually they put a lot of money try to target the Wnt beta-catenin signal pathway, and we also tried, you know, a number of, you know, you can name a number of different Wnt inhibitors. That was quite disappointing. So we didn't see any effects. So then we started to think about maybe we did something wrong. You know, we had a wrong beginning thought. We should, you know, get the right point. So so now we move on. We try to understand more about cancer stem cell biology, because initially we would think, you know, cancer stem cell might just hijack the property for the normal stem cells. So we try to, you know, use whatever we understood about normal stem cells and try to, you know, apply this into the cancer stem cells. But actually, they do have a difference. You know, they kind of different between normal and leukemia stem cells. But that's good. That's for the therapeutic purpose. That's really good, because even though for the gene expression profile, if you look into the gene expression profile, they have some overlapped gene signature, but they do have a different gene signature. So that's for the, you know, therapeutic purpose. That's very good. And now I think we try to understand more and more. So especially for all the cancer biology, it's very complicated. It's not really single signal pathway can drive this, you know, biology. It's like a multiple interaction, you know, crosstalk. So we have to understand this complexity and, you know, to develop the real treatment.
Perfect. Thank you very much, Jenny. Okay, we're going to move on to our next speaker. If you could give Jenny a round of applause. So our next speaker is Kevin Fleger. Kevin is an associate professor and head of molecular endocrinology and pharmacology at the Harry Perkins Institute of Medical Research and Centre of Medical Research at the University of Western Australia. We're changing pace a little. We're going out of cancer research into the whole area of receptor pharmacology. And Kevin and his group have developed a very, very powerful assay developed around bioluminescence that provides all kinds of data that is very complementary and goes above and beyond what most of the traditional assays that the pharmaceutical and biotech industry use to kind of understand receptor pharmacology. So I'll leave it to Kevin to fill you in.
Thank you. Is this okay? Everyone hear me okay? Okay, so take you through a little bit of my background. I did my undergraduate degree at Cambridge in natural science pharmacology, PhD in Edinburgh, and then 6 years postdoctoral training in Australia before becoming a head of the research laboratory in 2008. And so I'm now 15 years post-PhD and on my third nationally competitive fellowship. I have a number of hats, as well as being an academic. I was Chief Scientific Officer of Dimerix for many years, from 2008 to 2014, taking that through from early stage through fundamental research right the way now onto clinical trial with the company now on the stock market in Australia. I also chair the Australasian Society of Clinical and Experimental Pharmacologists and Toxicologists, which is ASCEPT. I chair their Scientific Advisory Committee in Australia, I'm a member of the AusBiotech, West Australia State Committee, and I chair Accelerating Australia, which I'll talk about in a moment. So my interests are understanding the molecular pharmacology of receptors, particularly GPCRs, so thank you very much for introducing those. And my particular disease focuses, although they can be very broad, and I've covered a whole range of areas, but in particular kidney disorders, cardiovascular disease, and cancer. And as was mentioned, my particular expertise is in bioluminescence resonance energy transfer. What is that? Well, I'll come to it in a moment. My world-leading expertise in that technology is very important because it can be done in real time and in live cells. So it's a very powerful profiling approach. And we can look at that for a whole range of receptor pharmacology. So if you're interested in profiling a particular compound, we can look at ligand binding, G-protein coupling, or GRAB2 if you're interested in receptor tyrosine kinases, arrestin, internalization, trafficking, and also this concept of heteromerization, which I'll talk about. So every facet of receptor pharmacology you might be interested in, we can look at in real time in live cells. And we also use a whole range of additional signaling assays for looking at proteins like inositol phosphate, cyclin-AMP, and ERK. So what is bioluminescence resonance energy transfer? Those of you who've seen the David Attenborough documentaries of those wonderful sea organisms giving off this bioluminescence, this blue light, or we can attach that to a receptor, our protein of interest. We can then have another protein we're interested in attached to a fluorescent protein. The importance of that is that when that luciferase and the green fluorescent protein come very close together, energy is transferred from the luciferase to the acceptor. We get less blue light given off and some yellow or green light given off, and therefore we can detect proximity. And it's very important in biology to understand how proteins are interacting, and therefore this is a very sensitive approach to enable us to look at that in live cells and in real time. We published a lot of this work in Nature Methods and Nature Protocols, established the first seminal method and protocol for this, and this is back in 2006. So this is using microplate readers, looking at this in live cells, looking at filtered light through the blue or the yellow channel. We've also developed, in collaboration with Promega, the nanobret ligand binding assays. So this was first published in Nature Methods in 2015. This is looking at binding using this same approach. And very similar to radioactive binding assays, but of course we're not using radioactivity. It's much safer. We're using bioluminescence. So we can do these experiments in live cells in real time for long periods of time. And unlike fluorescence resonance energy transfer, we don't have the problems of photobleaching. The other thing we can do, just another example, is look at trafficking. So in your cells, we have receptors on the membrane. Then they go into the cell through various different compartments and then cycle back up. And that's very important for their function. So we can actually test this by putting markers on the membrane and each of the different compartments. These are all validated with microscopy. We can actually track where those receptors are going in real time through the cell. And you can see this in real time. So this is over 2-hour time courses. And you can actually see from the different markers, they're going through the first compartment, the second compartment, and then through the various different compartments. So you can actually get time constants to get real-time monitoring this in live cells. We can also do this in multiplexing mode. So we can look at this in real time where we have the receptor tagged with a luciferase, which gives off blue light. We can then attach a yellow marker to one protein and a red marker to another so that we can actually truly multiplex, looking at 3 channels in parallel. And we can look at this again in real time. And this is illustrating another paper we've just published in Scientific Reports this year, where we've actually done this with CRISPR. And I won't talk about CRISPR, there are much better experts in CRISPR in the audience, but this is a way of gene editing that we can actually look at inserting the BRET tag at the end of an endogenous protein. So these proteins in cells under endogenous promotion, and they're actually able to be used for BRET. Receptor heteromerization, I touched on this a moment ago. What is receptor heteromerization? Well, we came up with a definition of this. There's a few people here that you may recognize if you're in the field. We came together at NIH to come up with a framework and a definition for this. And we came up with a macromolecular complex composed of at least 2 functional receptor units with biochemical properties demonstrably different from those of its individual components. A large complex, multiple different proteins. At least 2 of those proteins need to be different receptors. And importantly, those receptors change each other's function. So there's some kind of modulation which change what they do. Why is this important? Well, when we're doing drug discovery, we often run millions of compounds through, and we actually test to see what that does to our receptor. And we want to know what effect it has. But the point is that in our cells, we don't have receptors in isolation. Those receptors are, amongst other receptors, interacting with multiple different proteins. It's a complicated system. So if we can get to this in our cell culture, in our microplate reader, in a more physiological way, we can actually see that these receptors can transactivate each other, turn on other signaling pathways, give completely different pharmacology. What we've developed is a new approach to this called receptor hits, and this is patented, and we have IP granted in major jurisdictions around the world. The idea is that we actually have a configuration where we have a receptor here attached with our first reporter component of a proximity system, so like Brett, but this could be FRET, Tango, DiscoverX's system, any one of these proximity-based systems. We then have our interacting group, which could be a restin or G protein, that's attached to the second part of the reporter system, so it could be the acceptor of that energy transfer. And then we have our receptor that we're also interested in, but that is not labeled. So when our hormone or our compound binds to that receptor, the interacting group goes up and interacts. It brings the first and second reporter components into close proximity, and that gives our energy transfer. So this is very good for looking at intracellular signaling, but we can also look at ligand binding. So turn it upside down, and now we have a ligand binding assay which looks specifically at receptor heteromer complexes, but this time, again, in live cells in real time, and again, without the need for radiation. An example of this, so we have the angiotensin receptor system, which is very important for controlling